Okay, so I want to just apologize for not having been here yesterday. I got here only a few hours late. And my punishment is that I get to follow Ewan, which is a really hard act to follow. But he did actually introduce some of the things that I'm going to be talking about, so there is an advantage to that. And also, not having been here yesterday, there might be things in this talk which maybe you discussed yesterday. It sounds like there was a lot of talk about harmonization and some of the other aspects. But I'm going to try to bring in a different aspect. So I was asked to talk about data and IT challenges of achieving human cohort interoperability to drive new scientific discoveries and enable personalized medicine. So I'm going to talk about, what kind of data it is, what are the challenges, particularly harmonization, and facilitating access, actually finding the data in the first place. But what I'm going to do is bring in the low middle income aspect to this. So the East Africa project, I'm not going to describe the East Africa project because there's not enough time, but just some of the low middle income aspects to some of these challenges. So the data that I'm going to be talking about, so Ewan's already sort of distinguished between the health and the research data. So clinical data from EHRs is in the healthcare sector. In many countries, it's very well organized, whereas in low-middle income countries, sometimes it's still sitting in handwritten records. So it's not really a very good, easy resource to tap into, despite, in addition to all those legal challenges that Ewan mentioned. But then we also have these cohorts, and here you have in the research space where you have big cohorts working on specific diseases. You have omics data, you have phenotype data, you have biospecimens and metadata associated with that. So this is the kind of data that I'm talking about. So more specifically, a mix of phenotype data from demographic, anthropometric, disease and health related. And then on the omics data side, of course, you've got the whole microbiome space as well. But I'm going to focus a bit more on the genetic variation data because that's the kind of data that we work with. And that can come from various different sources. So you have your genotyping arrays, you have your whole genome or exome sequencing. And I mean, ultimately, you're looking at SNPs here. But one of the aspects I'm going to talk about a bit later is how comparable are those SNPs? So getting back to the data sharing issue. So what is required for data sharing? Well, you need obviously consent from participants. You need to have a robust data sharing model. So on paper, how do you, what MOUs do you sign? What data access agreements do you sign for sharing of data? But then you need the technical implementation. So do you transfer the data sets? Do you use the GA4GH streaming? Is it federated analysis? But the main problem, the main issue that I'm going to really discuss is the data interoperability. So curation and harmonization of data. It takes a huge amount of effort to curate data and also to harmonize it. But you also, of course, need infrastructure for data encryption. Human genetic data cannot just be transferred without encryption. The transfer itself, which many of you will take for granted, but in Africa, transfer of big data sets is a huge challenge. Storage and compute, submission to public repositories, but then also training in all different aspects of this. Very few clinicians get trained in how to curate their phenotype data and submit their data to public repositories. So the data sharing challenges specifically in Africa. So we've had a history of exploitation, they're vulnerable populations, there are a lot of cultural sensitivities. And so in terms of, that's studying the health side, but in terms of the science and sharing science, there's also been exploitation and what we call helicopter science. So people come in, they'll take samples, and they'll go away and analyze them. And if you look, so some studies have been done on the publications on African samples, and a huge majority, so 80% or more, do not have a senior author from Africa in sort of first or last authorship. So they're kind of losing their rights to science, their leadership of the science. This is starting to shift, but it's taking quite a while, and this feeling of exploitation is still there. I was at a meeting 2 weeks ago in Tunis, and I was amazed at how strong that feeling still is of exploitation from African scientists. We also have limited resources, access to resources, so to be able to analyze the data first and fast. So it gets very quickly exploited by other people before the African scientists or the low middle income country scientists have had a chance to really exploit that data. So the data are obviously anonymized, but there is a risk of identification. So when you're dealing with very small communities and everybody knows each other, you don't need very many variables to be able to actually identify a person. And then we have a big issue with ethics consent and what is actually allowed in different countries. So in East Africa, the ethics group actually did a study that took the ethics guidelines for 22 of the African countries that are involved in the H3Africa project. There are 34 involved altogether, but these are the ones they could get information on. Only 3 countries actually had legislation on how to deal with genomic data, because not many large-scale genomic studies have been carried out in many countries, so there was never a need to have specific national guidelines on this. Broad consent is actually not allowed in several countries. Some countries neither promote or prevent broad consent, and some countries allow for future use or secondary use of data, but it has to go back to the national authorities to actually approve that. So, and this is all described in this paper here. So not only are we dealing with what is the consent of the participant, but what is actually allowed at the national level. So there needs to be fair data sharing policies where there's a balance between protecting participants and the researchers themselves, and not having a barrier to advancing research and science and the potential benefits for health. So you need to maximize the availability of the research data, but protect the participants and the scientists, recognize the contribution of the scientists, look at the nature and ethics of the research, and then also make sure that this data gets deposited, but with a good custodians, so we have access committees that really protect the data and responsible use of that data. So some of the IT challenges, so those are some of the ethical challenges, but what a lot of you will take for granted, we don't always have in low middle income countries. So data-related skills for processing, analyzing, and interpreting the data, which means that the researchers don't have time to exploit the data, as I've said before it then gets shared and exploited by other people. We also have fewer computational resources, so there's only a handful of HPCs around the continent. Access to cloud is possible, but there are serious bandwidth issues in Africa. Data transfer and storage, so how many African institutions actually have secure storage and backup facilities where they have good management of data, encryption, secure management of data, and then obviously the bandwidth for transfer. So we found that it's actually cheaper and quicker to ship hard drives. So we encrypt data and ship hard drives around Africa because it's much cheaper and much quicker than trying to do downloads. So the next biggest challenge, which I'm going to sort of focus on probably for the rest of this talk, is harmonization and standardization of data. And I gather that you have discussed this quite a lot yesterday. So really, it's what is being collected, what is the variable that's being collected, and how is it being collected? So is it a measurement that's being taken where you get a number at the end? Is it a yes/no question, or is it a far more complicated question? And those variables, those values that you're collecting are not computationally comparable. One needs to do some sort of post-processing to get to the minimal standard of is it just a yes/no question? Do you smoke, yes or no? Or how many do you smoke a day? Or how many on average do you smoke a month? That the answer is not computationally comparable. So there's post hoc harmonization, which I guess cohorts here would need to do because a lot of the data has already been collected. But then one can also go back and start to build standards, which we've been doing in H3Africa. So what is the minimum data that should be collected for a particular cohort or for a particular disease, and how do you represent that? Can you map it to ontologies? Can you use Phoenix measures? And then that's the clinical and phenotype data, but then also on the genomic data. So if you run standardized pipelines and workflows, you should get the same result at the end. But if everyone uses a different variant caller, you will get different variants coming out. And as Ewan also pointed out, the reference genome builds. So you need to know that we've been working with the same build. We got a list of SNPs because we're designing the next version of our chip. And depending on how the data comes, you can or cannot find that, because sometimes you get a chromosomal location, but you'd never get told what build it was from. You get an RSID. RSIDs actually have changed. They don't match the chromosomal location. It is actually, as you pointed out, it's actually a much bigger problem than one thinks, being able to actually find and compare variants. So what is available? We have the Phoenix measures, which are measures for they kind of how to ask questions for phenotypes. We have ontologies, disease ontology, phenotype ontology, and then, as Ewan has so eloquently shown, the GF4GH standards. So how we're doing this in H3Africa is we've created a set of core phenotypes that everybody should collect. So I'll come back to that. We are mapping these, all the data that comes from case report forms to ontologies, so the phenotype and disease ontology, and then we're also mapping the ethics consent to this data use ontology to make sure that it's really clear what data sets can be used for what further studies. We've been looking at an ethno-linguistic ontology. I'll come back to that. For the genomics data, we're mapping to experimental factor ontology, and then biobank data is also being mapped to the Amayabus ontology. But then it's all very well mapping the data, but you have to make the data accessible and findable. So the data is all going to the EGA, and we're building a catalog of East Africa data so that the metadata itself is actually searchable. Because EGA, you can search, quite a simple search interface. In our catalog, you'll be able to search, I want a cohort from a particular country, studying a particular disease, where you have whole exome sequence data. And then you can actually go get the samples from the biorepositories and the data from the relevant EGA accession number. So this phenotype harmonization that we're doing, basically the phenotype harmonization working group that looked at all these projects, so they, at the time, there were about 30 different projects working on multiple different diseases, multiple different countries, and looked at what are the core, like the common phenotypes that are being collected. There were about 29 that were being collected, and then they tried to harmonize this. So they got all the data managers into a room and said, how are you asking the question? Went to the Phoenix measures, and some questions took an hour to really settle on what is the best way to ask a question. And sometimes these Phoenix measures had to be sort of switched, adapted a little bit to fit into the African context, so some of the terminology, for example. So we created a standard CRF for this, standard case report form and a data dictionary in REDCap. And we've mapped this to ontology terms. And then just going a bit further, one of the groups, so all the cardiovascular related projects, they came together and said, in addition to these, we have an additional set of phenotypes that we want to collect that's relevant to stroke, other cardiovascular diseases, so they could do full meta-analysis across these. But to do that, they had to actually develop some algorithms to kind of collapse to the lowest kind of common denominator of the data. So it's all post hoc harmonization, which is much, much harder. So now going forward for the second round of funding, a lot of the projects are using the standard CRF. So they will have a lot more data harmonization from now onwards. We also learned then extending this to other kinds of data. So there's the core phenotypes, adding pediatric questions, and then disease-specific questions, and then environmental questions, and then there's also the genomic data. So these are separate instruments, non-overlapping instruments in REDCap that people can just take and then build their CRF based on these standardized questions. So one of the issues that we've come across is ethnicity. So the standard phenotype looks at native language of mother and father and the ethno-linguistic affiliation. And there weren't actually enough, there wasn't enough standardization of this. So we've tried to build a bigger ethno-linguistic ontology, more specifically for Africa, but globally. And there have been a couple of papers. I know there have been many papers and talks yesterday about underrepresentation of the data, but here this is about how the data is represented. So these papers show that when you're trying to find ancestry data in the GWAS catalog or ClinVar, Africa is African. Africa is not one country. Africa is more than 54 countries, and yet the entire continent is represented as African or African American or admixed. So actually, we've been trying to extract data for microbiome studies and for precision medicine studies, and it's really hard to use simple terms to extract African data, so any data that comes from an African ancestry participant. So this brings me to kind of the last point of finding and accessing data. So not having the data labeled correctly or efficiently to be able to extract the data that you want, having ambiguous labels. So in our data set, if the data comes from the biorepository, country represents the country in which the sample was taken. If it comes from the genomic data set for the same sample, the country represents the country of origin of the person, which is fine if they didn't migrate, but sometimes it's ambiguous. Searching across multiple data sets, if we want to find all African data, we have to go to dbGaP, we have to go to EGA, we have to go to multiple different repositories. There's no single atlas catalog that has all this information. And then comparing genotype data. So are the arrays that we use compatible? Once you've imputed, maybe the data is more comparable. but the SNPs, the markers that are captured on arrays, are different depending on which array you use. Variant calling software, variant representation, that all becomes a problem when trying to immediately compare variants. Should you recall variants when you get a data set together? Joint calling, should you call it missing sites? So there's just a lot of issues around directly comparing variant data. So there are solutions. I've mentioned some of them, but really I think this like the data portal that was mentioned yesterday, I think would be extremely valuable for all people trying to do this kind of research. In East Africa, we're trying to make the data findable, accessible, interoperable, and reusable by doing mapping to ontologies, harmonizing, standardizing, getting it into the public repositories, and then mapping it to the data use ontology, so it's immediately obvious what you can use the data for. So just to sum up some of the key points then, there are a lot of challenges, not only in the data sharing part, but in equitable sharing of data. So when you're sharing data with a low middle income country, or with a group that doesn't have the resources you have, how do you make that collaboration equitable? How do you give good reasonable recognition to the scientists and get them involved in that process? They have the contextual information to interpret the data. So we need more training in data standardization and sharing. I'm sure it's global that many people don't learn how to curate data. I haven't touched on data integration and tools for discovery and decision support, but that's another challenge. But really we need to also think about making data findable, developing tools for access, making sure once it is shareable, is it interoperable, through standardization and harmonization, which is really, it's a hugely resource expensive undertaking to harmonize and standardize data. And finally, is it applicable? So there were discussions yesterday about polygenic risk scores. Can you extrapolate to other populations? No, you can't. A lot of the studies we're doing on African populations, we're finding that the allele might be in the same gene as has been found in other populations, but it's a different allele, or sometimes it's even in an entirely different location. And so what people have thought previously about some variants being pathogenic because they are at low frequency in certain populations, they're pretty common in African populations. So you need to look at the context information and think about what populations. You cannot just extrapolate to other populations. And having so little African data in the public repositories means we have such a much smaller resource to be able to go and validate and check in other studies. And also actionability and evidence. How much evidence is there for actionability of these variants might be actionable in European populations, but is that actionable in African populations? Is the action the same? And is that action even feasible? Is that test, is that therapeutic actually possible and available in Africa? So these are just a number of things one needs to think about in terms of some of the challenges. So thank you to the organizers for the opportunity to speak, and to the East Africa Consortium. Thanks.
I think we've got time for a couple of questions. Yeah.
Rory Collins, Oxford. Could I ask you about the challenge that you mentioned around equitable data sharing and this tension between, I think, what you referred to as helicopter science and the understandable enthusiasm of the funders, particularly, to make data very freely available to everybody, particularly in the context of prospective cohorts where it may take 10, 15 years of work before the cohort actually starts to become interesting, and then where you're having continual updates or follow-up. And how have you dealt with that? And not collaboration, because that isn't what is considered to be open access, which I think is what's referred to in your access policy. But the idea, how do you make it equitable for the people who put in 10, 15, 20 years of work in order to ensure that they do get a proper return?
Yeah, so I think obviously that applies to anybody who puts in 10, 15 years of work. So so these projects are funded by the NIH and the Wellcome Trust to have these requirements that the data goes into the public domain. So the data has to go into the EGA, and that the samples have to be available via 3 biorepositories that are funded. So the data access policy has been generated to be a bit more generous. So the timelines required for submission to public repositories is more generous than they would for other projects, so that there is more time to actually exploit and analyze that data. They have set up a Data and Biospecimen Access Committee who have, they're actually quite a good representation from Africa, so they're very conscious of the sort of African issues. And for biospecimens, there's a specification that for the first 3 years of request of access for biospecimens, it has to be in collaboration with an African researcher. For data, we can't enforce that, but it's encouraged. So the data access request form actually says, how will this benefit Africa? Have you thought about collaborating with an African researcher? But there is definitely a tension between the fact that you have to deposit the data, you have to make it available, it doesn't have to be collaboration really in the NIH and Wellcome Trust rules, and keeping that in line with the national laws. If the law does not allow for broad consent, you just cannot give broad consent. So you have to, so there's been a constant tension, and each project has to work with their grant officer to say what is actually feasible and allowable. So what we're trying to do is just say, okay, if the data is out there, can you try to collaborate with an African researcher? Maybe there's a capacity building component in there. And try to, I mean, everybody should be recognizing the people who collected the samples, who put all of those years and money into generating the data from that cohort. And Africa's not exempt from that. It's just that when you do have a partnership with an African institution, maybe that they cannot join that equitably in terms of the skills, the processing power. So you just need to. It has to be handled on a case-by-case basis, and it has to be done right. Otherwise, it's just going to further sort of build that gap, extend that gap.
Yeah, I totally understand the challenge if ancestry is crudely defined or coarsely defined as African. But on the other extreme, how are you reconciling what I know is an incredible complexity of underlying ancestral origins across the African continent, because you can't go to the other extreme either. So what, at a practical level, how are you folks sort of trying to come up with a framework that gives you enough information without making it almost impossible? So
we've been looking into this, with the change in the NHGRI policy about genome summary results having to be available. We're trying to work out, are these populations sensitive? And what we're trying to do then is use FST to group populations into regions based on the data, not necessarily geographical regions, to say at what level is there, can we group populations such that it's sort of less identifiable. So we might group into either known ethno-linguistic higher level groups, or northeast, southwest, central Africa. What we'll just try and do is with PCAs is actually use the data to tell us how best to differentiate better than African, but not as detailed as every tribal affiliation.
And what's the rough number of bins you're trying to categorize in? I mean, is it like 10, or is it more like 30 or 40?
Yeah, it'd be more like between 5 and 10. Yeah. But it'll also be coming out, what are we seeing in disease studies? Where is there differentiation in the population? Yeah.
All right, I think we've got time for one more.
Paul Elliott, London. It seems to me the consent issues that you raised could be a major constraint. I mean, how are you going to deal with that across different legal frameworks, different countries? I mean, there's one thing about access, but also if you cannot, if you have to go back each time.
Yeah, so there are two issues to the consent. One is the understanding from, is it informed consent? Do the participants who maybe have never had an education, can they understand what genomics research is? Then there's the country laws. And a lot of the country laws were there because they haven't had to deal with this before. So it's not because they had made a conscious decision to ban something. They didn't understand what it would mean. And so at several of our meetings, we've actually invited the ethics review boards from multiple countries and the people who are creating these laws. And they are starting to change. So since the paper was written, actually a lot of the countries have started to adapt their laws, now knowing that genomics research is bigger, it's the benefits for healthcare. So it is starting to change, and so it's a constant dialogue between the researchers, the funders, and these ethics and the national sort of legislation bodies to try and adapt. There will be some countries which won't change, but and we'll always have some sensitivities because of history, but it is being addressed.
Okay, we'll thank Nikki again for the interesting talk, and I think Jeff is going to talk to us.